Hello everybody, welcome back to our channel. We are on biology and we are on part B2.2. Before we talk about the cell membrane, the plasmic membrane, okay, now we talk about the organelles, okay, how the compartmentalization has been done in a cell. When you talk about organelle, of course, we have to remember it's only for eukaryote, okay, not prokaryote, okay. Organelle are discrete subunit in the cell, okay. So it performs a specific function, okay? And cell wall, cytoskeleton, and cytoplasm are not considered as organelle. No. The other are organelle, okay? Nuclei, vesicle, ribosome, and the plasma membrane are considered as organelles, okay? And study the function of the individual organelle. How can it possible? Because we have some advancement by ultracentrifuge and also by the cell fractionation, okay? This skill developed such that we can study the organelles in details. And the nucleus and the cytoplasm. First, we need to learn about the separation of the nucleus from the cytoplasm is important for compartmentalization. Okay. Why? Because eukaryote are cells with compartments and organelles. Prokaryote do lots of, but both of them has RNA, okay, and also has the process, okay. But for why is separated nucleus is important because they can separate the activity of gene transcription, okay. So they can have more time for the transcription. Okay? After transcription, they can do some translation, okay, some modification of the mRNA before they are released and meeting the represent the cytoplasm. Okay? But for the prokaryote, because they are not separated, okay, so the mRNA when they are being produced, they immediately meet the represent. No time for modification afterward. And also for the compartmentalization is important, okay, because they can have different function inside a cell, and some process of function even are incompatible. For example, for the phagocyte and the lysosome, they have to digest something. Okay, phagocyte have to digest the pathogens. Okay, lysosome have to digest the fungal cells and some invading viruses. Okay, when they do the digestion, they don't want to hurt the other part of the cell, so they have to engulf and surround the compartment of the pathogen, and the process of digestion can happen inside or without hurting the cell itself. Okay? And after the digestion, it is being released. So why we need compartmentalization? Because the process is incompatible. Okay? For example, in digestion of the pathogen and the digestion of the fungal cell. Okay? They don't want to hurt the cell itself, so it needs to be compartmented inside. And we go to an additional higher level, okay? But the additional level, we talk about adaptation, okay? How do a different compartmentalization adapt to their function? First of all, very simple, okay? Increase the surface area is important, okay? In mitochondria and also in chloroplasts, okay? Formation of a mitochondria from free prokaryotic cell, okay? For its model, because we don't look at the mitochondria is strange, okay? It has a double membrane. The other is single membrane, and it also has its own 70S ribosome, not the one in the other part of the cell, 80S. Okay, so we think that it comes from other places. Okay, so we think that we have a model. Remember, a large prokaryotic cell engulfs a smaller aerobic prokaryotic cell. Okay? And during the engulfment, the single membrane of the prokaryotic cell becomes a double membrane. Okay, inner membrane is from the cell, and the outer membrane is from the food vacuole. And the engulfed cell is aerobic, so it can provide energy and ATP. Later on, we talk about how it adapts to it. Okay, and the engulfed cell is not digest, symbiotic relationship. Okay, work together, so they fuse and become a mitochondria. Why we said so? Because we found out in mitochondria it has a 70S ribosome, not the outside one, 80S. Okay, and how the mitochondria evolve for its function are not of evolution about the compartmentalization. First of all, the task is to produce ATP, okay? First of all, it's a double membrane, okay? And the double membrane has a narrow intermembrane space, and this space is good, excellent, very reliable to produce the larger proton gradients to perform its function, okay? And then in the membrane is involved in the manifold, we call it a cristae, okay? Cristae is the important because increase the what surface area, okay? And also inside the mitochondria, it can have its own ATP synthesis, okay? And this thing can generate ATP from ATP plus PI effectively from outside of the mitochondria, just inside. Okay, and the electron transport chain can evolve to release energy later on from the reduce the NAD and reduce the FAD. Okay, and also to generate a proton gradient. And compartmentalization of the matrix with M site and substrate is evolved for the link reaction and the Krebs cycle. And not the thing you learn about the reaction later, but now you have to remember double mapping, proton gradient. Create manifold, increase the surface area. And that's all. And also adaptation for the corpus is similar. In the corpus, it has two kinds of reaction inside the corpus. One is light-dependent reaction and one is a light-independent reaction. They are different, so we have to separate them into two parts. Okay. One part is done is the tanacle. Okay, inside the tanacle, light-dependent reaction happens. So the chlorophyll is on the surface of it. Okay, and the tanacle is a stack of it stack it up become granum because increase the surface area, and different granum are connected by lamina. Okay, so all the light-dependent reaction is performed inside this part. Okay, and how about the light-independent reaction is outside, inside the corpus. Okay, it happens. Okay, we call it a Calvin cycle. Okay. And the adaptation of a corpus, okay, is of course a large surface area of the tanacle. Okay, and then making it a more surface area for reaction, and then the tanacle has a small volume of fluid, just small volume is important. Okay, and the compartmentalization of enzyme and structure of the Calvin cycle in the stroma is done outside of it. Okay, so we have different enzyme inside and outside. Okay, different reaction. Okay, because we have a compartmentalization in the two parts. Okay, so we can done it separated. Okay, first light dependent and then light independent reaction. And also I talk about the nucleus. Nucleus has a double membrane, so it's an evolution from it. It's important, okay, because inside the mitosis and meiosis, as a proliferate, okay, the mitosis and the meiosis, okay, the nucleus membrane has to be breaks down into several smaller vesicles and then split together, right? How can it be done? Because they are double membrane. The outer membrane is still there, and the inner membrane can break down into smaller vesicle. Okay, so you need to have a double membrane. The outside membrane keep the nucleus, and the inner membrane separate into smaller vesicle in the proliferation of mitosis and meiosis, cell division. Okay. And the nucleus, and also has some nuclear pore. Okay, and nuclear pore is important. Okay, to release and to absorb the mRNA. Okay, important. Okay, how can it be done? Because it's a multiple membrane again. So when the two double membranes are joined together, the pore appears. Okay. So double membrane is important because there are pores. Okay, there's some holes. Okay, in the nucleus. Okay, for the transfer out of the mRNA. And also, then we talk about ribosome. Okay, ribosome is another thing we will learn later. Ribosome has a free ribosome and bound ribosome. Ribosome is the place. Okay, it's a large subunit. Actually, the word literally is packages and smaller subunit. By these two units, okay, protein is synthesized from amino acid joining together. Okay, and this free ribosome is inside the cytosol. Okay, the fluid containing the cytoplasm. Okay, and then the polypeptides and protein are synthesized by the free ribosome, and then this kind of protein produced by the free ribosome is used inside the cell, except for the lysosome. Okay. And the other ribosome, not the bound ribosome, we produce a protein and polypeptide for lysosome, and also outside for exporting out. Okay, and then this kind of protein is used in mitochondria and chloroplasts. Okay, and then the bounded ribosome is another special features. Okay, bounded ribosome are ribosome attached to the organelle, the endoplasmic reticulum. When the endoplasmic reticulum has ribosome on it, it looks like rough. Okay, we call it rough endoplasmic reticulum. Okay, and the ribosome is still produce synthesized protein, but the protein is used to further develop by the first, okay, and it's used in the lysosome and it's used for secretion outside the cell, okay, not used in the cell, okay. And then how it happened also, it needs to be further processed by the Golgi apparatus, okay. So structure of the Golgi apparatus is a membrane bound organelle again, okay. Of course, it's eukaryotic cell, okay. It's located in the cytoplasm next to the endoplasmic reticulum near the cell nucleus. So cell nucleus, endoplasmic reticulum, and then Golgi apparatus are close to each other. It's made of a series of flattening stack of pouches called the cytosol. It's a higher surface area again. And what happened in the Golgi apparatus, okay? Because the Golgi apparatus, because the bounded ribosomes are produced. The protein has to be further processed and packaged to be used outside the cell or used by the lysosome, okay? So it's a further operation to mature, we can say, okay? Make the protein mature, make the lipid mature, okay? So after the ribosome produced the protein goes to the Golgi apparatus, and then after the operation, they is secreted out or they are produced in the lysosome. Okay. And how material is transferred within the cell, we remember by producing the vesicle. But how vesicle is produced from the cell membrane is by the cathepsin. Okay. So everything you learned already. This molecule are moved within the cell in vesicle, and then a single membrane is surrounding the molecule. Okay, cathepsin is used to coat the membrane of the vesicle. How the vesicle is formed, the membrane is formed is that cathepsin. You need to remember this name. Okay, and also remember vesicle can transfer the material because it can fuse it with the cell membrane, but the vacuole cannot. Okay. And this is the process, remember. Yeah, we learned it before, and then now we visit it, okay, because of the compartmentalization, the nucleus compartmentalization, the endoplasmic reticulum also compartmentalization, Golgi apparatus compartmentalization, everything is by compartmentalization. For different process to happen, okay, it's so clearly nucleus different process, producing the mRNA, and then of endoplasmic reticulum, producing the polypeptide protein, and then further development of the protein or the lipid in the Golgi. So at different parts, different organelle inside a cell have different function, and they have to separate it. And thank you for going through with me. I hope you can understand compartmentalization more, how important it is. Okay, the main concept is increased surface area. It is a main adaptation, okay, and separated different reaction inside, okay. Remember the photosynthesis, okay, light dependent reaction and the light independent reaction, and different and also the digestion doesn't hurt the cell, okay. Okay. So see you for next week for biology, okay. If you like our channel, like our channel, remember subscribe, like it, and share it with your friends, okay. See you next week, okay. Bye.